Hello, everyone, and welcome to today's The Scientist webinar. I'm Tiffany Garbett, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers, Dr. Jennifer Gommerman and Dr. Marian Pepper, will discuss their research on SARS CoV-2 antibody persistence and immune memory in recovered COVID-19 patients and the implications for protective immunity. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these questions as we can during our Q&A session. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors, Sinobiological. Sinobiological is an international reagent supplier and service provider with a comprehensive collection of SARS-CoV-2 research tools, which have been cited in more than 200 published research papers. Of all commercially available anti-SARS-CoV-2 N protein binding antibody pairs, Sinobiologicals is ranked at the very top for lateral flow assays. Ray Biotech. Ray Biotech is a manufacturer of proteomic research tools focusing on high throughput, high content, and ultra-sensitive immunoassays. Headquartered in Atlanta, Georgia, Ray Biotech is a full-service CRO offering collaborative research solutions to the scientific community. Biotechne. Biotechne empowers researchers in life science and clinical diagnostics by providing high-quality reagents, instruments, custom manufacturing, and testing services. The Biotechne family of brands creates a unique portfolio of products and services. Cayman Chemical. Cayman provides detection kits, screening tools, and antiviral agents to assist scientists studying SARS-CoV-2. Cayman's Coronavirus Resource Center brings all these resources together, giving you a way to easily navigate and discover the assets specific to your coronavirus research needs. Acro Biosystems. Acro Biosystems is a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics, vaccines, and diagnostic kits. Their comprehensive products include the high-quality, super-stable trimetric S protein, SRBD, S1, S2, and N proteins, mainly expressed by HEK293 cells, and paired antibodies with outstanding sensitivity verified by inactivated virus samples. PHCBI. PHC Corporation of North America markets a comprehensive line of laboratory equipment under the PHCBI brand. Products include space-saving and energy-efficient VIP Eco, Twin Guard, and VIP Series ultra-low temperature freezers, cryogenic and biomedical freezers, vaccine, pharmacy, and high-performance refrigerators, cell culture CO2 and multi-gas incubators, and Drosophila and plant growth chambers. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our resource list located on the left side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Dr. Jennifer Gommerman. Dr. Gommerman received her PhD in immunology at the University of Toronto in 1998. She went on to do a postdoctoral fellowship at Harvard Medical School, studying the complement pathway, and then joined Biogen Inc. as a staff scientist in 2000. During her tenure at Biogen, she became interested in B cells, multiple sclerosis, and the TNF superfamily of molecules. After 3 years in industry, she returned to academia as an assistant professor of immunology at the University of Toronto in 2003, and in 2015 was promoted to full professor. Gommerman's basic research focuses on how members of the TNF superfamily of molecules regulate immunity and autoimmunity. Her team uncovered a novel gut-brain axis that regulates neuroinflammation. With respect to translational work, Gommerman examines the role of B lymphocytes in multiple sclerosis patients, and she is the lead principal investigator on a study examining the effect of global migration on susceptibility to autoimmune disease. Since March 2020, she has also been studying the antibody response to SARS-CoV-2. Dr. Gommerman, let's just take a minute to make sure your slides are up and running, and we are good to go. Please feel free to begin.
Great, thank you. Can you hear me?
Yes, we can. Thank you.
Great. Thank you for the invitation for this scientist webinar. It's a pleasure to be here, and greetings from Canada. So I'm going to kick this off by talking a little bit about the antibody response to the virus. And I think that's going to set up Dr. Pepper quite nicely to then, by extension, talk about T-cell responses to the virus. But I want to start by thanking you, the audience, for joining me, because I know that this is a very tricky time to take an hour out of someone's day to listen to science. And we're all having to do this often from home while we're looking after others and taking Zoom calls and doing all sorts of crazy multitasking. So I really appreciate the time you've set aside for today's talk. And so, as mentioned already, I've spent a lot of my career studying B-cell responses, and these types of B-cells here are called plasma cells, and they make large quantities of antibodies. And we know that when infections occur, you get a wave of antibodies that are originating from plasmablasts and then plasma cells, and that these antibodies can persist for variable amounts of time post-infection. And that's what I'm going to talk to you about today with respect to SARS CoV-2. And I'm sure most of you are becoming painfully familiar with this diagram. This is the SARS CoV-2 coronavirus, and you'll notice that on the surface of the virus, there are these red spiky things. This is the spike glycoprotein, which is very characteristic of the SARS CoV-2 virus. And within that spike glycoprotein is an area called the receptor binding domain. And the receptor binding domain, which is on this spike protein, can bind to a host protein called ACE2, or angiotensin converting enzyme. So the SARS-CoV-2 viral structure allows for entry of the virus into epithelial cells that express this ACE2 receptor. And this structure is pretty similar to SARS-CoV-1, which is Torontonians will be very familiar with that virus. It emerged in Hong Kong, but also in Toronto in 2003. But there have been some modifications that the virus has made so that the receptor binding domain binding to ACE2 is actually quite strong for SARS-CoV-2. So SARS-CoV-2 initially infects the upper respiratory tract, so the nose and the mouth, and that's why wearing masks is a reasonable protective measure to take to protect ourselves and protect others from viral dissemination. And within these upper airways are these epithelial cells that express ACE2, but there's also other cells of the blood system, such as vascular endothelial cells, that also express ACE2. And based on the abundance of these receptors, you can get infectivity of the upper respiratory tract. And really, it's a combination. What we're learning is that viral dose is likely important, but also the host immune response and how potent it is early on in the infection makes a difference to how well a person will deal with this virus and the nature of COVID presentation. And I would refer you to some very recent studies that have come out that have looked at genome-wide association studies of patients who've had severe versus mild COVID, and certainly some genes are emerging that can make a difference in that host immune response early on. But the dose itself is probably important as well. So one possibility is that if you can clear this virus in the upper respiratory tract, that's likely a good thing, obviously, and it might even prevent the virus from really disseminating. And especially, we know when it gets to the lower respiratory tract and into the lungs, patients can have some real problems. And so we need to understand this initial immune response to the virus to figure out how the host copes with this infection. So our textbooks teach us that when we get infected with a pathogen, typically we first make an IgM response. So that's the immunoglobulin that's made by B cells. Before B cells are ever activated, they will be IgM producing cells. But when a B cell becomes activated, under some circumstances, it can switch out this IgM antibody molecule for other antibody molecules, particularly IgG and IgA. And our textbooks teach us that the order of presentation of these antibody molecules starts with IgM, followed by G, followed by A. Now, not all viruses behave this way, and it seems that SARS CoV-2 is not following the textbooks in that it seems that the IgA response actually comes up rather early in response to this virus, but then it crashes and you're left with very little IgA in the blood. And the same seems to be true for IgM. Whereas the IgG response comes up a little later, which we would expect. And from what we can tell, and I'll show you evidence for this today, this IgG response does seem to persist for quite a while in the host. Now, my lab is quite interested in studying IgA. This is an antibody that's made at mucosal surfaces, including the gut, where we make IgA in extremely large quantities, but also in the airways and in the saliva, and also in breast milk. So basically, mucosal sites, mucosal surfaces are places where IgA is made. And people often sort of ignore this antibody, but it might be pretty useful in dealing with this infection. And there's these 3 papers, 2 of which are preprints, that have come out recently showing that an IgA can actually provide some fairly good neutralizing potential against the virus. So usually we think about IgG antibodies in terms of their neutralizing capacity, and that's for good reason, because they are typically very good at neutralizing viruses, but we should not discount the other antibodies as well. All right, so why should we even measure antibodies to SARS-CoV-2? And this topic has been a little bit fraught in the media because we really would like to understand what is the scope of the pandemic. And I just want to make it clear that when you test for virus using the PCR assays that have been developed, you're only testing as a snapshot of whether you have a virus, you have the SARS-CoV-2 virus in your nasopharynx at that moment in time. But when you test for antibodies, what you're doing is you're looking for prior exposure to the virus. And I would argue that it's important to test antibodies simply because we need to understand more about this virus. What is the magnitude of the humoral immune response? It's another way of saying the antibody response. How much of it has neutralizing capacity? What correlates with neutralizing capacity? And how long do these antibodies last? So in March, when we ventured into COVID land, these were our big questions, as they were for many, many labs. Another good reason to measure antibodies, as I've already alluded to, is to get a sense of seroprevalence. So within communities, we know there are different burdens of viral exposure. So a person who's living in New York City is more likely to have encountered the virus than someone who's living in a rural area. Of course, that's changing. And so using antibody tests, we can, if the test is well designed, we can estimate how many people in a community have been exposed to the virus. And that's important epidemiological information. We know that, that there are vaccines in the pipeline and that they will be rolled out hopefully sooner than later. And so we're going to need a way of knowing how good vaccine uptake has been within different communities. And so using antibody measurements, we can tell whether a community has reasonable seroprotection as a whole by measuring antibodies levels. So there's lots of good reasons to measure antibodies, but there's also a lot of challenges. For example, in areas that have very low seroprevalence, you have a statistical problem in that it's going to be more likely that you'll have a false positive response in an antibody test. And obviously we don't want that, because if you have a false positive answer to an antibody test, a person might think they're protected when they really aren't. Also, most of the point of care devices that measure antibodies typically only measure one antigen. So that's what the antibody binds to. But this can be misleading. And what Deepta Bhattacharya has shown very nicely at the University of Arizona is that you really need to do more than one antigen to have an accurate assessment of seroprevalence. And of course, we know that antibodies, you get this big burst of antibodies when you're infected, but that will level out. That's normal. And so if we're going to do this properly, we need tests that are sufficiently sensitive to account for this natural decline in antibody titers that occurs. Now, I don't mean to imply that we're by far we are not the only lab that is interested in this, and this is a handful of different studies that have come out, whether they be posted up by preprint or or published, such as ours is, and I'll tell you about our study today, and you'll be hearing about Dr. Pepper's study as well, as she talks about T-cell responses. But the vast majority of the papers that have been published so far or posted have shown that the antibody response to this virus is relatively stable. And I'm going to show you some evidence of that in the slides to come. So when the pandemic hit Canada in March, we felt we were in a good position to start measuring antibodies because of a colleague that was down the hall from us had been studying SARS coronavirus since 2003 during the Toronto epidemic. And he had made, he was able to really quickly make beautiful proteins of the spike holotrimer, as well as the spike RBD and the nucleocapsid proteins. And these provided the perfect template for looking at antibodies against the virus in ELISA format. We also teamed up with my colleague, Dr. Anne-Claude Gingras at Mount Sinai Hospital in Toronto at the Lunenfeld Tenenbaum, and her postdoc, Kento Abe. And together they had set up a robotics platform for measuring antibodies in the serum. Now, our expertise, as I mentioned, is to look at mucosal antibodies. And so what we decided to do was use the same assay protocol that Anne-Claude was using, make some modifications, and try this on the saliva, and then meet back together at the end and ask how well did these antibodies correlate when they were measured by two different labs using two different biofluids. So I'm going to talk about the serum data first. So this is now published in Science Immunology. If you want to check it out, Isho et al. So basically, as I mentioned, the spike protein has this receptor binding domain that binds to ACE2 on a target host cell. And so if we can measure antibodies to spike and RBD as well as the nucleocapsid protein, we can get a sense of immunity to SARS-CoV-2. And that's what Anne-Claude did. She used these three antigens, spike trimer, and I think that's important, RBD and nucleocapsid. And then she took patient serum or plasma, but mostly serum, and detected IgGs, IgAs, and IgMs that were present in the serum and specific for these antigens. And the proteins were made by Jim Reaney at the University of Toronto, as well as the spike trimer was made at the National Research Council. And what Anne-Claude and Kento found was that they could detect a very nice signal in both acute patients and convalescent patients who had had COVID-19 but had recovered. And this signal was very strong for both spike and for the receptor binding domain, and was able to distinguish pre-COVID patients, and this was from a blood bank, a local blood bank that they used to look at pre-COVID-19 era serum as our negative controls. And you can see here that the acute response for IgG, there's still some that are negative, but this changes for the convalescent, whereas the opposite seems to be true of IgA and IgM, where they typically are reduced during the convalescent period. And the same was seen for RBD, and the two antigens correlated really nicely together. So that's good. We're measuring a synchronous immune response to this protein as well as the RBD. And for those of you who are aficionados in the audience in terms of how sensitive these ELISAs are, they were extremely sensitive, particularly for IgG. And it didn't matter whether it was run manually or on the robotic platform, the sensitivity was often above 98%. So that's really good. Now, one thing that, as I alluded to, is being a little bit debated in the literature is how long do these antibody responses last? And I think a lot of it has to do with the sensitivity of your measurements. So Anne-Claude used a highly sensitive chemiluminescent development procedure. And when she looked at antibodies to spike and RBD, and these are patients that were binned by time post-symptom onset, even as long as 115 days, which is as far out as we could go based on when we first started seeing patients emerge in Toronto, you see a very, very nice stable IgG response to spike, and you still see antibodies above background to the RBD, and pretty stable as well for the receptor binding domain. This is contrasted with the IgA and the IgM response that go pretty much down to the baselines by the time you reach 115 days. The other thing that Anne-Claude did, and she has made this work on her robotics setup, is she set up a competition assay to ask how well antibodies in these sera could compete for binding of RBD to ACE2. In other words, it's not a neutralization assay, but it measures antibodies as a proxy of neutralization. And what she found was that they could block ACE2 binding with these serum. In other words, these are presumably neutralizing antibodies, and that this signal was detectable up until the 106-day time point. We don't really know what happened here, but this particular group is not as well powered as the other groups. So now we're looking at some of the longer time points that we've started to accumulate from our clinical colleagues to see what's happening. But for the most part, the neutralization response is quite stable. Okay, so then entered our lab, and the reason, as I mentioned, we were interested in IgA because it's a mucosal antibody. And my a postdoc in the lab, a research associate actually, Dr. Olga Royas, who's just taken on a position as an assistant professor at the Krembil Brain Institute at the University of Toronto. In her previous life, she was working in Colombia as an MD PhD, and South America, that is. And she spent a lot of time measuring antibodies to rotavirus in saliva. So she was well positioned to supervise this project along with Gary Chow in my lab, who is the project manager and handled all of the very complicated REB and MTAs involved with patient samples and so on and so forth. And then these three students, Annie, Bauletta, and Michelle, who are all first-year graduate students, their projects were obviously interrupted by COVID, and they hadn't really gotten that far into their thesis work. So I asked if they wanted to join this project, and they were super game and did some really nice work. And then I want to acknowledge Mary Ostrowski, who is a clinical infectious disease specialist, works on HIV, in fact, trained with Tony Fauci, but was very interested in measuring this virus response in saliva and serum. And Alison McGeer, who's always on TV because she's been known since the SARS days of the Toronto epidemic. So, excuse me, we took saliva, and this is not as easy a biofluid to work with as serum. Because it's got all sorts of stuff in it, and it's got very high levels of IgA. But as I alluded to, it could be very important for neutralization. So we really wanted to look at this. And what we did was we took these things called salivettes that basically contain a small rubber, small cotton pad that you put in your mouth and you chew on it for a couple minutes, and then you put it into the sleeve that goes inside this tube, and then you spin it down, and it gives you a really nice clean sample at the bottom of the tube that you can then collect and use for your analysis. Our cohorts were acute patients from Alison McGeer from Mount Sinai Hospital, as well as convalescent patients from Dr. McGeer and Dr. Ostrowski at St. Michael's Hospital. And negative subjects were a little bit tricky because we don't have saliva banks. We have lots of blood banks, but not so much saliva banks. So at first what we did was we went to a rural area of Ontario where we knew that prevalence of the virus was extremely low. And we collected these samples and age and sex matched to the ones that we had from our colleagues. But eventually we did get some pre-COVID era saliva through collaboration in Saskatchewan. Now, one of our first challenges was if you're handling saliva, there's the possibility that it might be full of virus. And so we had to deal with this biosafety issue. And there are really two ways that the WHO has recommended for inactivating the virus. That is to do a detergent, Triton X treatment, or heat inactivation. And what we found was that if we looked at the titers of IgG or IgA to the receptor binding domain, we were able to retain that signal if we pre-treated the saliva with detergent, but heat inactivation really killed the ELISA signals. So that ruled that out in terms of a way to inactivate the virus. So we stuck with detergent. And we built this ELISA roughly based on what Anne-Claude had done in the serum, but we had to do quite a bit of trial and error to make sure that the ELISA was nice and clean and background was low. So the first thing is we incubated antigens on a streptavidin-coated plate. The titers in saliva are typically low. so not as high as what you would have in blood. And so what we found was we had to take advantage of an AVI tag within the proteins that were made by Dr. Rini, and then we used that, biotinylated the protein and plated it on these 96-well streptavidin-coated plates that were blocked. We also we wanted to pre-absorb the saliva against streptavidin, because streptavidin is a bacterial protein, and a lot of the antibodies in our saliva actually are used to seeing bacteria. So we wanted to make sure we weren't detecting anything cross-reactive to the streptavidin itself. So we pre-absorbed the saliva and then added it to the antigen-coating plate. And then we did typical ELISA steps after that. After that, it's pretty easy, and read out our ODs. And just for those who are interested, the way we did this, and this is not exactly high throughput, but I think it's a good way to do it, is that we took 3 different concentrations of saliva, starting at 1 to 5, 1 to 10, and 1 to 20. As I mentioned, titers in saliva are lower. And we took the area under the curve for our sample and compared it to what you would get for a positive control pool of saliva from COVID-19 patients. So in some cases, the samples we're measuring might have lower signals than that positive control or higher signals. It doesn't really matter, but we just basically use that to calculate an integrated score, which is a percentage, so that we run this particular positive control on each and every single plate. And what we found was that after infection, the total amounts of IgA and IgM were pretty stable and unchanged between negative versus COVID-19 patients. You might get a bit more IgG in COVID-19 patients compared to unexposed people, but for the most part, the antibodies are pretty stable. Compared to our negative controls, however, we got a very nice signal for anti-spike and anti-RBD IgG, as well as IgA and IgM. And the ELISAs were pretty nice and clean, like the background in the negatives was extremely low. So this was the time, at this point, we'd done enough patients, and then we decided, okay, let's compare our data with what Anne-Claude had in the serum. And what she found, we plotted her results on the y-axis and our results on the x-axis. And as you can see for the IgG, they correlate pretty nicely. So this means that the saliva, at least what we're measuring in the saliva, seems to reflect what's happening in the serum. The correlation is pretty good for IgM as well, not as good for IgA, and although it's still there. And we wonder whether there could be some local IgA immune response that's occurring that may throw off this correlation. But the correlation for IgG is quite good. So I guess the dream is, and it's becoming a reality because we're working on this now, is that if we're going to measure large populations, for the most part, I think serum is the way to go, or even dried blood spots if you're using a robotic setup. And I've already shown you how Anne-Claude set that up. But not everyone wants to give blood, and in particular, if you're going to do any serosurveillance among large pediatric populations and school-age kids, it can be difficult to draw blood. And so saliva might be a good alternative. And so what we're doing right now is trying to set this up with the robotic assay to do it at a 2 antigen level and to really get the sensitivity up a little bit by using chemiluminescence. And hopefully we'll have a reasonable way of looking at antibodies in large numbers of people, especially once a vaccine is rolled out. So that's my data, and it's just to summarize, COVID-19 patients make a durable antibody response to the spike protein in serum and saliva, and to a certain extent also the RBD. And this durability is particularly IgG. And the antibody response in the 2 fluids actually correlate nicely with each other. But I want to leave you with one last piece of data that's sort of buried in our paper because I think it's really intriguing. So this is our saliva data just shown a different way, where we've plotted each individual subject that have COVID-19 versus our negative controls. And you can see here how nice and clean this ELISA is when you look at antibodies to spike and RBD for IgG and for IgM. But look at what's going on here for IgA. We have a group of patients that seem to have, controls, and many of these are pre-COVIDs. These are people who've never seen the virus, some of whom are actually making IgA antibodies to the receptor binding domain. And we've since confirmed that this is something we can see in a separate cohort of patients of pre-COVID saliva from Norway, and we see the same thing. And so we're really interested in understanding whether these are pre-existing antibodies that recognize something else that could be potentially cross-reactive to the receptor binding domain of the SARS CoV-2 virus. So stay tuned for that. And this is the lab. I just want to particularly acknowledge Olga here and Gary, who really steered a lot of this work, and those 3 first-year students I was telling you about, Annie, Michelle, and Bauletta. Thanks for your attention. Oh, and funding from Ontario, the province of Ontario, and the Canadian Institutes for Health Research. Thank you.
Thank you, Dr. Gommerman. As a reminder to our audience, you can ask questions at any time using the Q&A tab located on the right side of your screen. Our panel will have a chance to address your questions after our next speaker. And that next speaker is Dr. Marian Pepper. Dr. Pepper earned her PhD in immunology from the University of Pennsylvania School of Medicine in 2006 and completed a postdoctoral fellowship in the Department of Microbiology at the University of Minnesota. In 2011, she joined the Department of Immunology at the University of Washington as an assistant professor and was promoted to associate professor in 2017. Dr. Pepper has authored 51 peer-reviewed publications, given over 75 national and international invited speaker seminars, and serves on numerous scientific editorial boards. Dr. Pepper's research team investigates how cells of the adaptive immune system form immunological memory by visualizing their differentiation, retention, and function in both mice and humans. The overarching goals of Dr. Pepper's research are to both enhance immune memory to design better vaccines and inversely block the formation of immune memory to prevent allergic disease. Dr. Pepper, let's just take a minute to make sure your slides are up and running. And they are. Please feel free to begin.
Great. Well, thank you very much for having me. And thank you to Jen for such a wonderful presentation and setup for some studies that we're going to talk about today, looking at the formation of immune memory to SARS-CoV-2. So I probably don't need to go through most of this with everyone, but just as a reminder of how immune memory functions, and specifically going way back to the to Burnett's theory of clonal selection, it's important to remember that cells of the adaptive immune system, meaning T cells and B cells, display these unique receptors that allow them to bind their antigen and become activated. For B cells, we call this the B cell receptor. When a B cell that has a specific receptor becomes activated, it will expand and differentiate so that it can work to control whatever pathogen it has just encountered. Because of this, and because of the necessity to have such a broad repertoire of B cell receptors or T cell receptors that can become activated, the body has billions of T cells and B cells that migrate throughout the blood and lymphatics, each expressing their own B cell receptor or T cell receptor, such that you have a large repertoire of potential receptors that can become activated. And what that allows you to do is to see a diverse array of pathogens so that you never encounter, hopefully, a pathogen that you don't have the right receptor for. But that also means that there are very few of these cells that are in any given individual. Otherwise, you would be overrun with lymphocytes. And so a very organized program of differentiation and proliferation needs to happen when an immune response gets going. And again, here is the B cell here that expresses the right B cell receptor for a virus. This time we're talking about SARS-CoV-2. When that interaction happens, you have this massive proliferation, exponential proliferation of these B cells as many clones of the same cell expressing the same receptor are made. And they also generate effector molecules. So for B cells, those effector molecules are antibodies, which are basically secreted B cell receptors. For T cells, these are cytokines or cytolytic molecules that can help to protect against the pathogen. But you don't want your body overrun with these cells that are making these potent effector molecules. So there is a sharp contraction of this response after the immune response controls the virus. And here you can see the viral load goes up. You start to make these effector molecules. The viral load goes down. And with that decrease in the viral load, you've hopefully been able to control the pathogen, but it's taken some time. So along with that can come fever, malaise, and illness, as there's this ongoing battle between the immune system and these pathogens. And once the adaptive immune response kicks in, hopefully you've controlled the pathogen, and that immune response can go back down to levels that are much lower. 90% of those cells will contract. But what you're left with are these remaining 10% of cells that there's about 10% that are over what you started with numerically, that have better qualitative functions that can control a pathogen far more rapidly. And just to demonstrate that, instead of this taking 7 to 14 days to control a pathogen where illness and disease can develop, an immune memory response upon a secondary exposure to a virus can happen within a day. If we do this with a bacterial infection, whereas it takes 5 days for what we call a primary immune response, this first exposure to a pathogen, to take place and control and start to express these effector molecules, it can take 2 to 4 hours for these memory cells to get activated and express these same molecules. These cells also migrate into different regions of the lymphoid organs. They also migrate into different tissues. So they're localized better, and they have differentiated such that they retain the ability to respond faster than they did during this primary response. And that therefore prevents disease. So obviously, studying immune memory allows us to understand how to make better vaccines, but it also suggests whether people are going to be protected from subsequent disease through a viral re-exposure. So when we hit March, we were studying all sorts of immune memory responses to various pathogens, focusing a lot on Plasmodium and other types of infections, but also on allergies. And we had this unique opportunity to pivot and to quickly start learning about whether immune memory can actually form in response to SARS-CoV-2 infection. And what we decided to focus on at that time point was really understanding if the vast majority of people who are exposed to SARS-CoV-2 that undergo mild COVID-19 disease, whether they induce persistent antiviral immune memory. And there were a lot of questions that were raised by this pandemic that we really didn't understand at the time. What type of immune memory would emerge? And by that, I mean, what cells would be involved? How would they function? Would they function like other viral specific responses that we'd studied in the past? And how durable were these responses? How long did they last? And this would obviously have important implications for individuals who'd been infected, whether they could potentially be protected from a secondary infection, and would also suggest potentially how better vaccines could be generated such that you could mimic this response if it seemed like it was a productive response. And again, things that I probably don't need to remind this audience about, there are many parts to the immune memory system. There are long-lived plasma cells that secrete IgG antibodies. These live in your bone marrow and produce antibodies. The wonderful thing about these cells is if they're continuously producing antibody and you get infected, you have antibodies on board that can prevent the interaction that Jen so nicely described between that RBD portion of the spike protein and the ACE2 receptor. And these are major mechanisms of protection for other viral infections like smallpox and polio and measles. B cells can also exist in the body that are memory cells that express B cell receptors and can get rapidly reactivated to generate plasmablasts, these antibody secreting cells. They proliferate a lot, produce a lot of antibodies. Those have also been shown to be protective in various viral infections. And then the memory CD4 T cells, which are near and dear to my heart, which really drive a lot of these responses. And we'll talk about this more in a minute, but memory CD4 T cells, some of these cells can interact with B cells to help differentiate the B cells. We call these T follicular helper cells. And they can also interact to help reactivate these memory B cells, while others produce cytokines, go out into the tissues and activate innate antiviral responses. So it was important to look at these cells as well. And really importantly, in cells that have, I think, been undervalued in this infection, maybe, the memory CD8 T cells, which can make interferon gamma and cytolytic molecules and kill virus-infected cells. So we wanted to look at this entire panoply of immune memory cells and really see what was there in an antigen-specific way. Could we find these cells, these rare cells that emerge, and could we track them over time in individuals? And what we were lucky enough to be able to do was to pull together a cohort of individuals who had recovered from mild COVID-19 in late February, early March. And we knew that these were all PCR positive. About half of them had been at the same event together. We compared the immune responses in these individuals to 17 healthy controls. And these were individuals that had not had SARS-CoV-2. We tested their antibodies by ELISA and compared them to historical negative controls. That's what this HN here is, which were samples much like Jen described that were pre-2019, so they hadn't had SARS-CoV-2. And what was really nice about this is that we set it up as a longitudinal analysis. So we brought the same people in roughly about 9 to 10 days after they had been exposed, and then another 10 days after their PCR test. So it was about 20 to 40 days for that first blood draw. So the average was 35 days. And then we brought the exact same people back about 2 months later so that we could look again to see how their responses had changed over time. And this is currently under review. It's in a preprint that you can find on medRxiv. And without going into all the details that are in that medRxiv preprint, we can say that we first looked to see if acute inflammation had been resolved at this early time point, which it largely had. There were a few cells, memory B cells, that looked like they were still contracting from the early phase of the response. But largely, the immune response looked normal. It looked like it had returned to homeostasis, that those lines that I showed you before had gone through that contraction phase. And at this second blood draw, we further confirmed that looking at markers of cell cycle analysis, Ki67, different markers of activation. And so these really were memory time points that we were looking at these two different time points that we were able to analyze. The second one being more quiescent than the first. So I'm just going to go through what the different cells that we looked at were, and I'm going to go through the antibody data really quickly because I think Dr. Gommerman's research really, the two of us reflected each other's results quite nicely. So we were curious to see if long-lived plasma cells emerged, and one of the ways that we could tell that long-lived plasma cells had emerged is if the antibody response is maintained, but the cells that make the early antibodies, the plasmablasts, are gone. So we confirmed that the early plasmablasts were completely gone. There were no plasmablasts left, which are short-lived effector cells. But when we looked at antibody titers, this time against the RBD specifically, and this is from the plasma of these individuals, we could see at both visits, the RBD titers for IgG were higher than the healthy controls, and importantly, they were maintained across the entire time course. And as Dr. Gommerman just showed you, we saw very similarly that the IgM and the IgA antibody levels declined, which to us suggested that plasma cells that secrete antibodies into the blood, long-lived plasma cells that secrete antibodies into the blood, were still making IgG, but we had less of these cells making blood-borne antibodies for IgM and IgA. Now, I think Dr. Gommerman's hypothesis that maybe the IgA cells are in another site, it's really intriguing and an important contribution. So we also demonstrated similarly that these plasma antibodies could inhibit the interaction between the RBD portion of the spike protein and ACE2 using these surrogate virus neutralization tests. We also went in and showed that with a real viral neutralization test in the BSL-3, that the levels of antibodies that could neutralize at these late time points were largely maintained. So even though some of the antibody isotypes went down, the IgG levels were retained, and these were still able to neutralize. So we next looked at these IgG Memory B cells and looked to see if Memory B cells that had been associated with protection from other viruses were also generated in these mild COVID-19 convalescent individuals. And just for a quick primer, for those who don't think about T cell activation and B cell activation and how all these different cells meet up, when a B cell gets activated by binding
Hi, Dr. Pepper, if you can hear us, we've lost
Hi, Dr. Pepper. Sorry for...
We're just going to connect you in a few minutes.
Sorry, Dr. Pepper. Yes, it looks like we lost your audio. You may need to reconnect, and we'll have to share your screen again here. Sorry for the technical issue, everybody. We'll be back in just one moment. Hi, Dr. Pepper. If you can hear me, you should just have to click on that microphone icon to share your computer audio.
Okay. Sorry about that. I don't know what happened. Hopefully I'm back on.
Loading now.
Can someone confirm that you can hear?
Yes, I can hear you.
Okay, great. Thank you.
Sorry about that. We're just getting it back up and running for you.
Okay.
I think we're good to go.
Okay, great. And I don't know where it cut off, but I will just jump through this for the sake of time. And we were talking about the formation of germinal centers and how you can find cells that have undergone these processes of class switch recombination, where they change their BCR isotype, somatic hypermutation, where point mutations are introduced into the B cell receptor. And these cells that express these new B cell receptors are then tested for their antigen binding ability. And those that bind well to antigen are then able to get survival cues that leads to a competitive process and better binding of those B cell receptors in a process called affinity maturation. So all of this is happening over time. And although people have looked at germinal centers in response to immunizations and see germinal centers form and then disappear within 10 to 20 days, during an infection, these germinal centers can actually last for very many weeks to months. Throughout this process, you get new B cell receptors with these mutations introduced and potentially enhance affinity over time. And the output of these germinal centers are these memory B cells that I was just talking about that are able to respond to challenge, and also the long-lived plasma cells. So we wanted to look to see if we could find memory B cells. We already showed that we could find antibodies that suggested that long-lived plasma cells were there, but could we also find these memory B cells? And were they going through this process in which their BCRs were becoming mutated and looking like they were gaining affinity? And so to do this, we took a trick that we developed in Mark Jenkins' lab by Justin Taylor, where we made B cell tetramers where we would basically conjugate a biotinylated RBD protein to a streptavidin that's also conjugated to a fluorophore, in this case, PE. And we've done this with several different SARS-CoV-2 antigens. We've done this with both RBD and spike, but RBD was what we focused on for these studies. We also use a decoy reagent because B cells are really good at binding the PE molecule, the biotin, the linker, all of these different components of the tetramer. So we come in, we bind with our decoy reagent first to identify any B cells that are specific for components of the tetramer. And then we come in with our tetramer itself to identify the rare antigen-specific cells. Because these cells are so rare, we need to do an enrichment of these cells. So after we do the decoy binding and the reagent binding, or tetramer binding, we add anti-PE magnetic beads, and then we enrich those cells that are labeled with the PE molecule over a magnetic column so that we can then elute our cells of interest, which have been enriched through this process. And that's important to note because it's important to understand what our plots look like. Here on the y-axis, you have the decoy. I'm sorry, that came off. And on the x-axis, you have our tetramer. So in these gates, you can see these rare tetramer positive cells. And these are in the SARS-CoV-2 positive individuals, while these are the cells that can bind our reagent that are in the healthy controls. And while there are some B cells that can bind our tetramers in the healthy controls, a lot of these B cells looked a lot like naive cells. There were a few cross-reactive memory cells in here, but there were a lot more of these RBD-specific cells that were found in our individuals who had undergone infection. And this was especially true at this late time point, suggesting that those long, prolonged germinal centers were potentially still releasing memory B cells into the blood that we could then pick up at these late time points. And we characterized what these cells looked like based on different markers, specifically memory B cells that are sort of classically defined, express both CD21 and 27, which you can see in this upper right-hand corner, but there are different variations of activated B cells that express different levels of CD21 and 27. Naive B cells largely can be found in this bottom right-hand quadrant. And you can see that in our healthy controls, the bulk of the individuals who had RBD-specific memory B cells, we're really looking at naive B cells here, whereas these individuals who had experienced disease had far more of these classical memory B cells defined by double positive expression of CD21 and 27, and then some of these other cells, some of these other phenotypes as well. And so you can see there's a big increase again over the healthy controls of these memory B cells, especially at visit 2 from the CoV-2 individuals. I also said that activated B cells that undergo a germinal center are also associated with class switching. We could compare the isotype of these B cells by looking at IgG and IgA and IgM and IgD. We could look at all the different isotypes on our B cells of interest. So this is looking at RBD-specific memory B cells and then looking at the level of class switch cells. And the vast majority of the cells in the CoV-2 individuals didn't express IgM or IgD, whereas those in the healthy controls did, again, associated largely with naive individuals. When we further broke that down, we could see that the bulk of those class switch cells expressed IgG, while a smaller population that varied quite a bit between individuals expressed IgA. And that's just showing this here. For the sake of time, we'll just keep going. We also showed that these memory B cells are quiescent, but they expressed low levels of the transcription factor TBET, which has been associated with reactivation in a variety of viral infections, suggesting that these cells are poised to respond rapidly to a reinfection. So we decided to test whether their B cell receptors had the capacity to neutralize the virus at these late time points, suggesting that they could be protective, and also wanted to look at their levels of somatic hypermutation. So we single cell sequenced RBD tetramer positive cells. This is just showing the post sort of our individual RBD specific cells. We could sort across all of the RBD specific cells and then look at cells with a variety of phenotypes. So we could look at whether they express CD27 and 21 or whatever other markers we wanted to look at. But what this allowed us to do is to single cell sequence each of these individual B cell clonotypes and look to see if their B cell receptors were somatically hypermutated, and B, if we produce those antibodies if we produce those B cell receptors as individual antibodies, what could they do functionally? And so this is just showing some of the heterogeneity of these different responses. This is from the person that I just showed you. In the last slide, the vast majority were 27 high. And 21 high, whereas in this individual, we had some that were 21 single positives, while some were double positives, and same with this one. There was some variety here, and now we can actually go back and see if there are differences in function of these antibodies. But it was important to see if the antibodies that were directly isolated from these various memory B cells could actually inhibit binding of the virus to the ACE2 receptor. And so first we showed that of all of the antibodies that we sequenced and expressed as IgG monoclonal antibodies, every single one that we isolated with our reagent could bind IgG, so that was encouraging. These others are negative controls, and a positive control is in here as well. We then did inhibition assays, and we showed that some of these at the first time point could also inhibit the interaction between RBD and ACE2. We then went into repeated this exact same thing at the second visit, and we could see again the specificity of our reagent is really strong. All of our antibodies were RBD specific, and some of these and even more of these could neutralize at this late time point. And this is showing what those same viral neutralization assays look like in a real BSL-3 viral neutralization assay. And at both time points, we had very good levels of viral neutralization from the B cell receptors that were isolated from our memory B cells. So this really shows that these RBD-specific memory B cells expressed B cell receptors that could neutralize the antibody. We went on to also test their affinity by BLI and also look at their levels of somatic hypermutation. They have low levels of somatic hypermutation, somewhere roughly around 2 to 4%. That increased over time, consistent with longevity of the germinal center. And most importantly, these B cell receptors were capable of neutralizing virus. So what about the T cells that are required to help these B cells, the CD4 follicular helper cells? And also, interestingly, what about the CD4 effector cells that activate the innate cell responses? I'm going to go through this data more quickly, but I highly suggest you all look at the paper itself to look at this in more detail. But what we decided to do to prevent the reactivation of cells through activation assays, where sometimes you can see activation of cells because naive cells have been incorporated into the response, what we decided to do was to actually sort various populations of memory cells directly from the PBMCs from our donors. And we divided up the PBMCs gating on CD4 T cells into central memory cells or effector memory cells that do not express the CD45RA molecule, but express varying levels of CCR7 versus naive cells. And we then put these sorted populations into culture with autologous monocytes as antigen presenting cells and spike protein and a little bit of IL-2, and looked to see if there were memory cells that could proliferate to a high degree in those populations, and if that proliferative capacity was greater than in the naive cells, showing that this truly was a memory response. And so what you can see in the naive individuals, these are the healthy controls. If you took naive cells from a healthy control and you put them into these culture assays, very few of these cells proliferated within that 5 to 6 day time point. If you took them from the CoV-2 individuals, we had a small population of cells that could proliferate. We think that those might be contaminating from the memory pool because obviously you don't see that here. So it's unique to the CoV-2 people. But importantly, what we could see is if you sorted central memory cells or effector memory cells from the CoV-2 convalescent individuals, we saw these really robust populations of proliferated cells that were spike specific. And you didn't see this in the vehicle. You saw this in the spike activated populations. And importantly, they expressed the chemokine receptor CXCR3, which is associated with Th1 responses in humans and interferon gamma production. About 20 to 50% of these cells also express CXCR5, suggesting that they are memory TFH cells, which was exciting. And this is just showing the increase over vehicle. This is spike minus vehicle in the healthy controls or the SARS CoV-2 patients in red here for naive central memory and effector memory cells. So again, without having the time to go into all the details, we showed that there's spike-specific circulating TFH cells in these individuals. We showed that upon spike re-stimulation, they could express receptors associated with B cell activation, including ICOS and CD40 ligand. We showed they also express cytokines, and we found that for the non-TFH effector cells as well. Most of the cytokine that was produced by these cells was interferon gamma and some IL-2. Very few of these cells produced IL-17, but there were some individuals that had a few.
Your screen sharing has been
more.
Hi, Dr. Berger. We've lost your screen. We can hear you perfectly, but we've just lost your screen. Hang on just a second.
Okay. We're having all sorts of fun.
Let's try to reshare your screen for us.
Okay. It says sharing on. Should I stop sharing?
Let's bring up your presentation.
Okay.
And then just go to presenter mode there. Perfect. Thank you. We're good.
We back?
We're back.
Okay. We're going to get through. We're almost there.
We're almost there. That's fascinating. Thank you.
Okay. So we haven't been able to talk about all of the different things we looked at, but what was really nice about these studies is we could look at the same individuals over time at as many different types of adaptive immune memory responses as possible. So we looked for antibody production associated with long-lived plasma cells. We looked for memory B cells and their expression of B cell receptors that were capable of neutralizing the virus. We looked for spike responsive cytokine producing CD4 T cells and ones that could actually help B cells by the expression of these receptor paired ligands. And in data I didn't discuss today, we also looked for CD8 T cells, which was interesting in the way that we looked at CD8 T cells, which was interferon gamma production. We actually didn't really see much of a difference between CD8 T cell responses from the healthy controls versus the CoV-2 individuals, which may suggest that those CD8 T cells that we're looking for might be actually in the tissues themselves and in the lungs themselves at this time point, but it raises some really interesting questions. So a lot left to do, and we're still working with these same individuals to see how long this memory response will last. The latest time point that we looked at for some of those individuals was 110 days after they had symptom onset. So now we're going out further to see how much longer these cells are going to persist, and are they going to stay in this stable phenotype. Obviously, these were all individuals who had mild disease, and that was the goal of this study, was to really understand those with mild disease. But what about people who haven't had mild disease? What does that look like, and what's the comparison? And there were some studies early on and more recently suggesting that individuals who had really severe disease lost their circulating TFH cells. So looking at those individuals and whether they come back would be really interesting. And of course, the one thing that we can't definitively say is, are these individuals who have these robust multifunctional viral specific immune cells, are they going to be protected? And hopefully we can get into that as time goes on. We'll be able to track that a little bit more. So this was a massive collaboration between the University of Washington with my lab. These studies were headed by Lauren Rada. You can see here, a postdoc in the lab, Jason Netlin, a research scientist. Mike Gale's lab was incredibly helpful and did all of the organization with the IRB and helped us with all the blood collections, sample collections. And then Julie Eggenberger did the vast majority of these neutralization assays in the BSL-3. Neil King and the Institute for Protein Design was amazing and generated a lot of the proteins that we use for our reagents. This was a close collaboration with David Rawlings' lab. They did a lot of the sequencing of the B cell receptor on our memory B cells. Jess Hammerman's lab worked out a lot of the early studies showing that the cells were quiescent at the time points that we looked at and had returned to homeostasis. And Dan Campbell's lab did those nice sorts of the memory cells versus the naive cells to look for antigen-specific proliferation. And just to thank all of our funders who have allowed this study to happen, as well as all of our volunteers who were so wonderful and agreed to do this important study. So with that, I will take any questions.
Wonderful. Thank you both so much for your presentations. As a reminder to our audience, you may ask questions at any time using the question box. The audience has already submitted several questions, so let's get to them. Our first question is for Dr. Gommerman. How do you expect the antibody response that you have measured will compare to the response a person might experience when vaccinated?
So first of all, can you hear me?
Yes, I can.
Great. Okay, so that's a really good question. So as you saw, there was pretty much everybody makes an IgG response to this virus. It might take time, like we see it more in the convalescence than the acute, but the IgG response is made in virtually everyone. But there is, of course, some dynamic range within that response. And the goal with a vaccine is to really trick the immune system to make a response that's quite strong. So, for example, we know that there are some people who make a misdirected immune response, in particular on the T cell side. So, vaccines can potentially get us around that problem and make sure that the antibody response and the T cell response are appropriate and strong. To do that, there's a number of tricks that vaccine developers use to really stimulate the immune system in a strong but safe way. And that's why we think vaccines are the way to go moving forward, so that we can, because we're never going to get there from herd immunity, that's for sure. Mm-hmm. And we're going to need these vaccines to make sure that everybody who can make a strong immune response is protected.
Great, thank you. Our next question is for Dr. Pepper. The loss of protective immunity in more severe cases are often the elderly. Could it be related to the aging of the immune system?
Yeah, I think that that's definitely a possibility. Obviously, as the immune system ages, there's less thymic output, there are less T cells to direct the differentiation of these B cell responses. So that could decidedly be part of it. And then, of course, you have that layered on top of whatever's happening in the loss of innate cell function. So I do think that probably has a very important impact on how these different responses develop.
Great. Dr. Gommerman, did you get false positive results from people that may have been infected with SARS CoV-1 in your data?
We only had one subject that we knew for sure had SARS CoV-1. You got to remember that SARS CoV-1 was relatively contained quickly. So the chances of having people who've had SARS CoV-1 in these studies are pretty low. But there was one subject that we knew had it. And their antibodies, even though they were negative for SARS CoV-2, did bind to the antigens that we measured. But it's important to keep in mind that we didn't necessarily look at the neutralizing capacity for those antibodies. So we did see some cross-reactivity from this one person, yes.
Interesting. This question is for both of you because you saw very similar results, and we'll start with Dr. Pepper. How long do you think the IgG antibody response will last in SARS-CoV-2?
Yeah, it's a great question, and something that we're still looking at. I think studies after ours, this preprint first came out in the beginning of August. So there have been studies out to at least 6 months that still show that there are IgG antibodies present. So we have yet to know exactly, because unfortunately we're limited by the time since this virus emerged here. But so far, so good at 6 months, and we'll keep looking to see what changes.
Dr. Gommerman, would you like to add anything to that response?
Yeah, no, I would have, my answer would have been the same. The earliest patients are from Wuhan, and there is a study from Wuhan showing that the antibody response is durable to at least 6 months.
Okay, interesting. Dr. Gommerman, researchers are discovering that the SARS-CoV-2 virus in wastewater. How is virus shedding in feces or in urine correlated with antibodies that might be found in either the serum or the saliva?
Yeah, we don't know. I think, if we had been super organized when we collected saliva, we would have also asked for stool, but I think my REB would have just blown up. But I think it's a cool question, because it's possible that if you can make a brisk antibody response at the site of infection, that maybe there won't be that kind of dissemination, but we just don't know.
Great. Dr. Pepper, do you expect to see sort of an incremental response in the antibody or the immune response for patients that might have mild COVID-19 infection, severe COVID-19 infection, or somewhat intermediate responses?
Yeah, it's an interesting question. I think I think there's a little bit of a Goldilocks principle with the immune response. I think you want it to get activated adequately to engage these various cell types and have them differentiate appropriately. But I think with severe disease and severe inflammation, we know that can really trip up some of these very well organized programs that need to happen. So I'm not sure that it's going to be incremental in that way. And I think we need to really study some of the individuals who've had severe disease to see what their immune memory looks like.
Interesting. On that same note, do you expect to see antibody response in perhaps individuals that are asymptomatic, Dr. Pepper?
Yeah, and I should have mentioned this. Some of our cohort really didn't have any severe symptoms at all. And so I don't know how we're defining asymptomatic completely, but didn't have a fever, didn't have a cough, just felt a little malaise. So they were probably included in that. I think it would be interesting to do this. One of the things that I think would be really interesting to do this study, to repeat this study, is in kids or at least young adults who've experienced this with absolutely no symptoms whatsoever. And I think in our cohort, the average age was 30 to 40. So it would be interesting to look at individuals at a younger age, less than 18, and see how they're responding.
Indeed. Dr. Gommerman, do you think antibodies against other parts of the virus would show identical patterns of immune response, or would each response be unique?
So I think the question is, from person to person, do antibodies target different parts of the virus? Is that the question?
Or instead of looking at, let's say, the RBD region or the spike protein, would it be different if you looked at other areas on the virus as opposed to those target areas?
Yeah, so we did do, and Claude, my colleague, did some work on the nucleocapsid, and you get a nice response against that as well. However, it tends to be a little more cross-reactive, so you see more signal in the negative subjects. So we have looked at other antigens. I think for example, if you think about the Regeneron treatment that has one very famous patient that has taken it, that's a combination of two monoclonal antibodies, and the rationale for that was because the receptor binding domain is a fairly it's small, but it could potentially put out escape mutations and evade the immune response. And if you look at their paper, the paper that Regeneron published on their combinations of antibodies, and they looked at a whole bunch of different combinations, they found that they really needed two antibodies, two monoclonals to put the brakes on the virus. So when we get exposed to the virus, or if we're exposed to a vaccine, we will make a polyclonal antibody response, and that will cover a variety of places on the virus and should give us protective immunity. So hopefully that answers your question. It's the difference between a monoclonal response and a polyclonal response. But when we're exposed to the virus, that's what we make, a polyclonal response.
I see. So a very similar question, and this would start with you as well, Dr. Gommerman. Have you seen any antibodies or any antibody response that might be detrimental in your studies?
We haven't looked at I think the questioner is referring to antibody-dependent enhancement, and we haven't looked at that. Marion, I haven't seen really much literature around ADE. I think if we had seen this as a big signal, it would have come out in the literature, I think, already. I'm not saying it's not there. It's just I'm not seeing a huge evidence of this in the literature.
I agree.
Interesting. Dr. Pepper, there is some discussion that T cells might be more useful for vaccines due to potentially longer lasting immunity. What are your thoughts on this? Do you think that the T cell response might perhaps be longer lasting?
I think first of all, it's probably important to to remind everyone that the T cells and the B cells really function together in a lot of different ways. B cells provide a lot of survival cues for T cells. T cells obviously help differentiate the B cells. So the hope is that you have all of these cells being maintained for long periods of time. I don't think from our evidence so far that there is any good data showing that the T cells outlast the B cells. If anything, our memory B cell responses were increasing over time. So I think that hopefully all of those cells will stick around for a while. But I think that's the ideal situation.
I can just jump in too. I think it's also important. It's also important to recognize that we're only limited to the biofluids, the saliva and the blood, and I guess we can also look in the stool. But and so if you're looking for cells, cells have a habit, especially memory cells, of going to live in tissues. This is certainly true for both memory B cells and T cells. So there were some studies looking for memory B cells years after SARS infection, and couldn't find them in the blood, but that doesn't mean they're not in the body. And so I just want to throw out that caveat because it's, unless we were to take a bone marrow aspirate or other things, it's really hard to know. These cells, as memory, they're not as useful in the blood as they are in tissues where they can quickly respond to reinfection. So that's something to keep in mind.
Interesting. So on that note, Dr. Gommerman, why did you choose to look at saliva as opposed to plasmid or serum?
Okay, so mostly it was just practical. We had serum available, but some of the early measurements that Claude did were done in both serum and plasma and got similar results. So it was more just a convenience thing. Interesting.
Dr. Gommerman, you showed some fascinating results at the very end of your presentation where you see a IgA response in the uninfected individuals. What do you think that might be due to? Some research studies are suggesting that perhaps common cold coronaviruses might produce an immune response. What, if you're willing to speculate, sort of what might that be due to?
Sure, yeah. So in terms of the T cell response, there's a recent paper that shows that this sort of notion of pre-existing immunity, which I think people have measured in the T cell compartment as being anywhere from 20 to 50% of unexposed pre-COVID blood samples. A recent paper has come out to show that that is very likely other circulating coronaviruses. But what we picked up in the saliva were IgA antibodies to the receptor binding domain. And there's no way that that was because of SARS CoV-1 from 2003, because there just weren't enough people to justify that. And we've seen it in another cohort from another continent where SARS CoV-1 was not present. So I don't think it's because of the receptor binding domain is quite unique from SARS CoV-2. I don't think that this is necessarily circulating anti-coronaviruses that have induced this antibody response. So we don't know what it is, and we're certainly looking, and we don't know what it means either. But just to point out, though, that most of those antibody titers that we saw were lower than what you saw in a COVID-19 patient, although some of them were getting up to about 50% of what you would see in the COVID-19 patients. So, yeah. I guess we'll just have to see how that plays out.
Interesting. This is a bit of a speculative question again for both of you. And we'll start with Dr. Pepper. Given what we know about the current mutational pattern and rate of SARS-CoV-2, what is the likelihood that recurrent vaccinations will be required?
That's a great question. I mean, I think we've all been encouraged by the slow mutation rate of this virus so far and hope that continues. I think I'm right now just focused on getting to that first vaccine. I don't know, Jen, do you have anything that you want to add to that?
Yeah. I mean, I think the first wave of vaccines are going to be different than the second wave because the first wave are mostly mRNA or adenovirus-based vaccines. And the second wave are, I think, there's more protein subunit vaccines coming down the pipe, and that's what we're used to using for flu. And I think that the protein subunit vaccines are going to be pretty good for inducing antibody responses. So I think, it's going to be also a question of not just do we make a durable immune response, but, is the vaccine of choice or the vaccine that one takes going to be enough for that? Or are we going to need a boost? It's really too early to tell. And normally this process takes much longer than 10 months. So, we'll see what the first vaccines give us and whether we hit it lucky and are able to make a good durable immune response from those vaccines, or whether it's going to take something down the line to do that is unclear right now.
Interesting. So you both showed that the IgG antibodies persisted longer. Dr. Pepper, why do you think that might be? Why do the IgG antibodies persist longer than the IgM or the IgA? And what might be the functional significance of them persisting longer?
Can you hear me? It just said that my connection was lost, but
yes, I can hear you.
Great.
Great.
I think Dr. Gommerman got to this a little bit earlier. What we are sampling is the blood. And so what we're capable of sampling is the antibody that is secreted by long-lived plasma cells in the bone marrow. And if that's not where the IgM and IgA memory B cells or plasma cells, we don't know which yet, but if that's not where they are secreting their products, then we're not going to measure it. So I think that's going to be part of the answer.
Okay. Dr. Gommerman, would you like to add anything?
Nope.
Okay, great. And now we have time for just one more question, and I'm going to direct it to you first, Dr. Gommerman. What would you say is the implications of your research for persisting immunity or long-term immunity for SARS-CoV-2?
Well, I think that our research, Marion's research, and many other research papers that I cited in the beginning are a good news story for our battle with COVID-19, because it implies that, first of all, if you're infected, chances are you will never be reinfected. And we've now had 40 million people infected with this virus and only a handful of cases of reinfection. So that, I think, is in and of itself good evidence that we make strong and appropriate immune responses to this virus. However, I would caution against any hope for herd immunity, because we know that the seroprevalence is still very low, and people, not everyone makes an appropriate immune response, and certainly people die from this terrible virus. So I think, but the good news is that I think this means that a vaccine is definitely plausible and should work and should institute this same type of immune response, if not even better, to really put an end to this.
Dr. Pepper, how would you summarize your results for the larger scope of protective immunity?
Yeah, I'm not sure I can add much to what Jen just said, because that was exactly what I was thinking. But I would say that our studies show that you have a broad range of immune cells that are behaving the way we would want them to protect against reinfection. And again, I think that this is encouraging and is suggestive that we will be able to get a vaccine that will be able to get us beyond this pandemic, hopefully.
Indeed. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. Jennifer Gommerman and Dr. Marion Pepper, as well as our sponsors, Sinobiological, Ray Biotech, Biotechne, Cayman Chemical, Acro Biosystems, and PHCBI. Thank you, everyone, and have a great rest of your day. Goodbye.